Welcome back to my channel. So today we will look at grade 12 life science, meiosis. So before starting the chapter, we'll go through some basic terminology, right? So the first thing that you have to know is the definition of mitosis. What mitosis is. So mitosis is a type of cell division in which one parent cell divides to form two genetically identical daughter cells, each with the same number of chromosomes as the parent cell. So the purpose is for the growth and repair. So let me give you an example. So when we get a cut on our finger, basically cells divide by mitosis to repair the tissue in your finger and for healing to take place. So do you remember sometimes when we get cuts in our body and then after a week or so it gets better on its own? So it's mitosis doing its process to heal the tissue. Then parent cell, like that's the definition or that's the word that we mostly use in mitosis. So what is a parent cell? Parent cell is the original cell that divides to form new cell. Like it's in the name, parent. So it's the original cell that divides to form new cells. Daughter cells are the new cells that are produced when the cells divide. So parent, it's the original, and daughter cells are the new cells that are produced, right? Alright, now you have to know the definition of meiosis. Like, it's a must. Knowing the definition of meiosis and mitosis is a must. What is a meiosis, right? Meiosis is a special type of cell division that halves the number of chromosomes, right? So it's a division that halves the number of chromosomes, and then 4 genetically different haploid daughter cells are formed from one diploid cells. So some basic information that you have to know is mitosis. So mitosis occurs in the body cells, also known as the somatic cells, and meiosis occurs in the sex cells, that is also known as the gametes, right? Now let's dive into more complex terminology. Chromosomes. You have to know the definition of chromosome, guys. So chromosome carries genetic information in form of genes, right? So this whole here is a chromosome, and this part here is a chromosome, right? So there are 2 chromosomes here, right? There are 2 chromosomes. Then chromatid. Chromatid is one of the 2 identical copies of chromosome after the DNA has been replicated. So this is a chromatid, this is a chromatid, and this is a chromatid, and this is a chromatid. Centromere. Okay, so centro meaning, centro meaning center. So centromere is where 2 chromatids of a chromosome are held together, which means this part here is a centromere. And you should know all the 4 of the definitions because they usually ask an exam not to define it, but they will put this structure there and then they will ask you, which part is a chromosome, which part is a chromatid, which part is a centromere, like that. And then, homologous chromosome is a pair of chromosomes having the same shape. This is having the same shape, same size, and having the similar genes for each characteristic occupying the same position. Homologous chromosome meaning this one might be from the mother and this might be from the father. So homologous mean one from the mother and one from the father. That's it. Alright, then we have bivalent. Bivalent meaning a pair of chromosomes that are physically in contact with each other at a point where crossing over will take place, meaning bivalent. Bi means 2, which means they're together. So which means that it's a homologous chromosome that are physically in contact with each other at a point where crossing over will take place. Then we have again, this replicated and unreplicated chromosome, they don't really ask about it in exam, but it's just better for you to know. So unreplicated chromosome has a single double-stranded DNA molecule. There you go. single double-stranded DNA molecule, then this replicated has two identical double-stranded DNA molecule. Quite simple, right? And then moving further, interphase is the the phase in cell cycle where DNA replication takes place. So we learned this during meiosis, right? So during the interphase, DNA replication takes place. Diploid, meaning 2 complete set of chromosome in a cell. Haploid, 1 complete cell of chromosome in a cell. Gene. So you should know definition of all this. Gene, a segment of DNA in a chromosome gene codes for a particular characteristic like hair color, eye color, etc. For example, somebody says that, oh, somebody tells you that, oh, your hair is so good and it's so thick. And then you reply and say, oh, it's just my genes because my mom has the same hair, my grandma has the same hair. And for example, eye color, like most of you, our eye color matches with our mother or our father or our grandmother. And, you say that, oh, it's genes. When somebody says that, oh, you're genetically blessed. So it's like that. Genes, they code for a particular characteristic, like your hair color, eye color, etc. Moving on. So now the complex stuff are coming in. But don't stress, we will look into it. Alright, so centrosome. Centrosome is an organelle containing 2 centrioles found in animal cells. Let's look more into centrosome because we like to ask about centrosome a bit. So centrosome is an organelle found in animal cells that serve as the main microtubular organizing center. It plays a key role in cell division, especially mitosis and meiosis, by forming the spindle fibers that pull chromosome apart. Structurally, the centrosome usually contains 2 centrioles at the right angle to each other. So that is a centrosome. And then, guys, we have then we have centriole. Structure formed when 2 centrosome divides into 2. They move to the opposite end of the cell during cell division. So, okay, let me try to zoom in. Okay. So you see this. Okay. You see the structure, right? It has T inside, right? So when you look at it, it should instantly click in your mind that, okay, this is a structure. It has T inside, which means that what? It is centriole. I think, maybe in exam, most of the structure will not show a T inside that, but you should just keep in mind that, the inside part here, this part here, it's a centriole. Just keep that in mind. Alright. Crossing over, very important, is overlapping here of homologous chromosome resulting in exchange of genetic material. So the process or the function of crossing over is to exchange genetic material. Then we have the chiasma, point where 2 chromatids overlap during crossing over. So the process is crossing over, but the point is chiasma, however you like to pronounce. Then karyotype is a representation of the number, shape, and arrangement of a full set of chromosomes in a nucleus of a somatic cell. So this thing here is a karyotype. Then autosomes, the first 22 pairs of chromosomes which control appearance, structure, and functioning of the body, which means that this 22 are the autosomes. Then the gonosomes are the sex chromosomes that determine your gender, whether you're going to be a male or a female. XY, it's female. XX, it's male. Okay. Moving on. Somatic cells are the body cells. Sex cells are called the gametes. Alright. Non-disjunction is when a homologous chromosome pairs fail to separate in meiosis during the phase anaphase, right? Then we have karyokinesis, which takes place in the nucleus. Karyo in the nucleus, and cytokinesis takes place in the cytoplasm. Meaning karyo, nucleus, cytoplasm. So karyokinesis is the division of nucleus of the cell. whilst the cytokinesis is a process whereby cytoplasm of a single cell divides into two daughter cells. Like, they don't basically ask about cytokinesis and karyokinesis. I haven't seen much of those questions, but it's just good to know. Moving on. This is the interesting stuff. So first of all, you should know this permit. Okay. So P it's for prophase, M it's for metaphase, A it's for anaphase, T it's for telophase, right? Then prophase 1. First we should know the structure of it. So meaning this part here is a chromosome. With a T it's a centriole, and this one is a spindle fiber. So during prophase 1, I'm going to send you the slides so that you can read it thoroughly, but I'm just going to briefly explain you what each part is about, because here I think we have not so important information also. So during prophase 1, what basically happens is that the process of crossing over takes place to bring about variation, right? So why this crossing over takes place? To exchange the genetic material. So what basically happens during prophase 1 is the process of crossing over takes place, then nuclear membrane and nucleus starts to disappear and forming of spindle fibers. So the three main things that happen during prophase 1 is nuclear membrane and nucleus starts to disappear, spindle fibers starts to form, and the process of crossing over takes place. As you can see here, this is the before, this is the during, and this is the after. Now guys, I told you that this point, I told you that there's a name for this point, right? So what is it? If you said chiasma, it's right. And what's the process? So the process is crossing over. So in an exam, they will provide you with a structure like this, and they will ask you that what process has been taken place. Then you can say that the process of crossing over has taken place. And how do you know? You can know that this two colors has interchanged, which means that the process of crossing over has taken place. Then we have the metaphase. Okay, here is we have a lot of information, but we don't need all of them. So during metaphase, what usually happens is that homologous chromosome move to the middle of the cell. So homologous chromosome move to the middle of the cell. Basically, it's the equator, and the chromosome lie on the opposite side of the equator parallel to each other. So homologous chromosome align at the equator, and then random arrangement takes place to bring out variation. So the two things happens here. Homologous chromosomes align themselves at the equator, and then the second point is the random arrangement takes place to bring about variation. Anaphase, nothing fancy happens here. What happens here is one whole chromosome from each pair is pulled to the opposite poles by contraction of the spindle fibers. In simple words, you can say that the chromosomes move to the opposite poles. As simple as that. You don't need to write this fancy terminology. If you want to write that, go ahead. Then you can go ahead. You can get same mark if you write the same thing. Moving on. Telophase. So telophase is the nuclear membrane forms around a group of chromosomes at each pole. So you can see that this is a nuclear membrane forming around a group of chromosomes at each pole. Nucleus returns, cytokinesis takes place, splits the mother cell into two daughter cells. Right? Important. Each daughter cell now has half the number of chromosomes, and each has a slightly different genetic makeup due to crossing over. To look at the second meiotic division. So the second meiotic division takes place in both daughter cells formed during the meiosis 1. So prophase 2, what usually happens is that again the nuclear membrane and the nucleus starts to disappear, centrosome splits into two centriole, and spindle fiber starts to form, and then the chromosomes are not in there. So pretty much everything, it's the same from prophase 1, beside the fact that prophase 1, the chromosomes were in pair, and now during prophase 2, they are not in pairs, right? And then metaphase. So during metaphase, what happens is that single chromosomes align themselves at the equator. So during metaphase 1, homologous chromosomes align themselves at the equator, but then during metaphase 2, single chromosomes align themselves at the equator. Moving on, anaphase 2. Centromere splits and then the two chromatids are pulled to the opposite poles. Meaning here, the two chromatids are now pulled apart and then moved to the opposite poles. And then we have the telophase 2, in which a nuclear membrane starts to form around unreplicated chromosomes at each pole. You can see here the nuclear membrane is forming. And then cytokinesis splits the cell into two new cells. Moving on. So this is the summary I made about the meiosis. So you could just read it for your exam, take a screenshot, or download it from the slide, and it will be beneficial for you for the exam. Like, you don't need to read all this information and know every single word for them. Like this here, it's would suffice. So let's go through it. So prophase 1, what basically happens is the nuclear membrane disappears, spindle fiber starts to form, and crossing over takes place to bring variation. Metaphase 1 again, homologous chromosomes move to the equator of the cell, then they lie on the opposite side of the equator parallel to each other, and then random arrangement takes place to bring out variation. After that, we have anaphase, meaning one whole chromosome from each pair is moved to the opposite poles. And for telophase, nuclear membrane forms around a group of chromosomes at each poles and formation of two cells. Then for prophase 2, nuclear membrane disappears, spindle fiber starts to form, and chromosomes are not in them. So it's one and the same thing as prophase 1. It's just that crossing over does not take place and chromosomes are not in the pair. Otherwise, it's one and the same thing. For metaphase 2, chromatids align at the equator and each chromosome become attached to the spindle fibers. For anaphase 2, chromatids are pulled to the opposite poles when the centromere splits. And for telophase, nuclear membrane forms around the unreplicated chromosomes. Cytokinesis splits the cell into two new cells and the formation of four genetically different daughter cells. So that's it for the meiosis. Then okay, this you have to know if you want to make it easier. So permit again. So P it's for pair up, M is for middle, A it's for apart, T it's for terminal. Now let's look into the process of crossing over. So why is crossing over important? Because crossing over brings about an exchange of genetic material during the process of gamete formation, which results in the formation of new genetic combination. And this results in gametes that would give rise to individuals that are genetically different from their parents and siblings. In simple terms, crossing over leads to variation. It brings about exchange of genetic material, resulting in formation of new genetic combination, and leads to formation of gametes that will give rise to individuals that are genetically different from their parent and sibling. After that, we'll now be learning like so much about meiosis. So we have to learn the importance of meiosis, like what is it important. So the meiosis importance is production of gametes for that the cells produced, halving the number of chromosomes from diploid to haploid so that the chromosome number remains constant from generation to generation. The other one is a mechanism to introduce genetic variation through crossing over during prophase 1 and random arrangement of chromosomes at equator during metaphase 1 and 2. Okay. Now, what's the difference between meiosis 1 and meiosis 2? It's quite simpler. So for meiosis 1, chromosomes align themselves at the equator in homologous pairs. Here they align individually. Here the whole chromosome moves to the opposite pair. Here the chromosomes move to the opposite pair. Two cells are formed at the end of the division. Four cells are formed at the end of the division. The chromosome number is halved during meiosis, and here it remains constant. Crossing over takes place, and crossing over doesn't take place. Um, After that, let's see the difference between mitosis and meiosis. Mitosis takes place in somatic cells, body cells, while meiosis takes place in sex organs or gametes. So here, for mitosis, both karyokinesis and cytokinesis occurs one. For meiosis, it occurs twice. For mitosis, two daughter cells are formed. For meiosis, four daughter cells are formed. And daughter cells are genetically identical to one another and the parent cell. Here, they're genetically different from each other and from the parent cell. Chromosome number remains constant. It's out. Crossing over doesn't take place during mitosis, but in meiosis, it does have place. The only similarity between mitosis, prophase 1, metaphase, and anaphase, and meiosis 2 is prophase 2, metaphase 2, and anaphase 2, which means that they don't have mitosis, don't have telophase. Right, and there I made a mistake. So XX is female, XY it's male. Right, guys? So now let's try to answer this question. It says that the diagram below represents part of an abnormal human karyotype. So how many autosomes are shown in the diagram? So the autosomes are the first 22 chromosomes in the karyotype, then the chromosome is the 23rd one. Then the autosomes are responsible for your physical appearance, characteristics, etc. And the chromosome is like the function is to determine sex, whether they are male or they are female. So they say that how many autosomes are shown in the diagram? 1, 2, 3, 4, and 5. So the answer would be 5. Okay. They are not, sorry. So they're not asking how many autosomes are in the entire karyotype. No, they're not asking that. They're asking how many autosomes are shown in the diagram, which means there are 5. Then they say that the name the type of chromosome represented in the pair number 23. What do you call this? So I just told you that it's called a gonosome. Right? Moving on, they say that name the disorder represented in the diagram. So guys know that whenever they ask about a disorder of a karyotype, it is Down syndrome. So that's going to be Down syndrome. And process during anaphase of meiosis that resulted in abnormal number of chromosomes in the karyotype. So the process is then non-disjunction. So now that the process is non-disjunction, but the disorder is Down syndrome. The last question states that what's the gender of the person presented in the karyotype? So you can see that it's X and Y. So it's going to be a male. If it was XX, it would be a female, but this one is a male. So the question says that the diagram below represents a chromosome pair undergoing a process during meiosis. So question 1.5 says, name the organ in a human male where the meiosis occurs. So in a male is testes, and in the female it's ovaries. But here they are just asking for male, so you just write testes. They say that process represented in the diagram. So you can see that here the two homologous chromosomes are overlapping each other, which means the process is crossing over. And then they say that label the area P. So I told you that the point where they touch each other or the point where they overlap is called chiasma, right? Structure. Okay, what's the structure Q? It's in the center. You can see that they're asking for the center. So if they ask for a center, it's what? Centromere. Of course, they're asking for the center. Next thing they're asking for is name the structure R. Where is the structure R? Is this one single strand, and which is the chromatid? Yeah, that's it, guys. So the next question is, how does non-disjunction leads to Down syndrome? Note that non-disjunction is the process. Down syndrome or trisomy is the disorder. So what happens is when chromosome pair 21 fails to separate during anaphase, non-disjunction occurs, and one daughter cell receives 24 chromosomes. So if it separates, then it is going to have 23 chromosomes. But if it does not separate, it's going to have 24 chromosomes, and that's what we are learning about. And when this gamete is fertilized by a normal gamete containing 23 chromosomes, the resulting zygote has 47 chromosomes, leading to Down syndrome and also known as trisomy 21. The next question is saying that explain the process of crossing over, and they usually like to ask this ask this question in exam also. So the process is very simple. So what happens is during prophase one, homologous chromosome lying next to each other, chromatids from each homologous chromosome overlap at a point called chiasma to exchange genetic material. This is the process of crossing over, and it's very simple. You should just memorize this and write it in exam, and you can easily get 4 to 5 marks. That's it, guys. And then the next question is, describe the role of spindle fibers in the movement of chromosome during meiosis. They also like to ask this question, and it's not tricky, it's like too simple. So the spindle fibers shorten to pull the chromatids to the opposite poles. That's it. That's the function of spindle fibers during meiosis. So you just have to know this, and you'll be good to go.